So I'm Marina Dutra Clarke. I'm a genetic counselor at UCLA. And how we're going to set up this talk is I will start by giving you an overview of genetics, and then I'll hand it off to Dr. Bianca Russell, who's a clinical geneticist, and she'll share some really interesting cases, and you'll see how we've applied a lot of these genetics principles. And our talk is called The Genetics Primer for the Modern Nephrologist. And this talk was supported by Natera. So we know that genetics plays a role in nearly all aspects of medicine, but a particularly strong role in kidney diseases. And Dr. Rastogi just highlighted the many benefits of knowing the underlying genetic cause of the patient's kidney diseases. And so it's very important. I really want to emphasize that today. We've actually been in this genetic revolution or genomic era for quite some time now, though. We've sequenced the human genome 20 years ago, and next generation sequencing was developed 10 years ago now. So I think by now we really should be offering genetic testing routinely for these patients. And you've probably noticed how fast-paced the genetics field is. There are tens of thousands of genetic tests on the market, and with more options, it can be more confusing in selecting the right test for your patient. You've probably noticed that the list of genes on panels can vary between commercial labs. The test methodologies can vary between labs. So if possible, collaborate with clinical geneticists and genetic counselors to help select the appropriate test for your patient and for the right price for your patient. So after the sample's at the lab, how do they analyze the sequence data? Well, the lab is looking at your patient's variants, which are the sequence changes in the genetic code, and they're taking those variants and comparing them to the variants in our large data sets of other people in the general population data sets. So, and looking to see what is different in your patient. The problem with this is that there are millions of variants that differ between one individual and another. So human variation is normal. So it's very difficult to decide whether a variant is benign or a disease-causing variant. Most labs use this 5-point scale that indicates the likelihood that the variant is disease-causing, ranges from benign to likely benign to variant of uncertain significance or VUS, likely pathogenic and pathogenic. And then I listed out some of the common tools and criteria that's used in variant interpretation and classification. So for example, population databases are key. Like if the gene is typically associated with a severe kidney disease and it's highly penetrant, then we would not expect that variant to be commonly seen in our population database. Also, is it the right variant for that gene? So for example, some kidney diseases are exclusively caused by a particular variant in a particular domain of that gene. So is it the right mechanism? And then has it been reported in the literature before? Has that variant been seen before? Is there co-segregation data? So often we'll order genetic testing for other family members and see, is that variant tracking with the disease in that family? And also, does the clinical and family history fit with your patient? You know, sometimes phenotype is not part of the variant level interpretation, so definitely use your clinical judgment and always send in your clinic notes to the lab. And what about the in silico prediction tools? What do they say? Those are the computer algorithms that can help predict the effect on protein function. And are there any in vitro or in vivo experiments for this particular variant? Because that would be very helpful too. And this is also why genetic test reports are 10 pages long sometimes, because they lay this all out for you, all the databases that they used and all the tools that they used. Hopefully in the future, our patients will have better access to orthogonal methods like RNA sequencing and proteomics, but for right now, you're pretty much working with this list. And something to really keep in mind is that some labs don't agree on variance classification. So there is discordance. Even though they're all using similar guidelines, like standardized guidelines, there's still discordance. So here's an example. There was a study that came out last year where they took 8 labs, submitted variants, and their classification, and nearly half of those variants had discordance between 2 or more labs. And 63% of the pathogenic and likely pathogenic variants were discordant with variant of uncertain significance by another lab. And 11% of the discordance cases would have impacted clinical recommendations. So here's a sample genetic test report that I want to walk you through. So starting from left to right, the gene is listed, PKD1 gene. And the disease that it's associated with is polycystic kidney disease. And the inheritance is autosomal dominant. So we have 2 copies of all of our genes. And autosomal dominant inheritance is where there's a pathogenic variant in one of those gene copies. And then here in the variant column, that lists out the GPS coordinate of your variant. So here it's always given in 2 nomenclatures. So the top line is the cDNA nomenclature, and the bottom is what's happening on the protein level. So the cDNA, that's showing you which variant is changed. In this case, the variant was deleted. And then there was an effect on the protein. So we have the leucine is the first protein that's affected by this. And it's changed to a tryptophan. And in fact, this deletion of that 1 base pair caused a frame shift in the entire code. And it affected all of those amino acids, 93 amino acids further down, and then led to a premature stop. So FS stands for frame shift, and then the asterisk tells you that there was a stop. So this is a truncated protein product. And it's heterozygous, meaning they found that variant in about half of that sample, so consistent with that autosomal dominant inheritance. And the lab classified this variant as pathogenic, and they list out the rationale under the interpretation section. And I just outlined some key points in the interpretation section showing that it is the right type of variant, because as a frame shift truncating variant, that does fit with the mechanism for PKD1. And also it's been reported in the literature in unrelated individuals. So that's showing replication. And then lastly, the variant is absent from our Broad Nomad dataset. So this variant is rare and not seen in our healthy general population. So now I've just explained this to you. How do you explain this to the patient without using technical terms and technical jargon? So I would just, I would keep it simple. I would just say, our genes are the instructions for the body to function properly. And one of those genes is the PKD1 gene. And there was a typo found in that instruction. So the 11,554th letter was deleted in those instructions. And that caused the instructions to stop short. And this is called autosomal dominant polycystic kidney disease. So in addition to just giving the patient the genetic information, what else would be important and part of the genetic testing, genetic counseling process? Well, genetic counseling is the process of helping people understand and adapt to the medical, psychological, and familial implications of genetic contributions to disease. And genetic counselors really like to emphasize the part that says and adapt. So it's not just giving them the genetic information. We have to also help our patients adapt to this information. So work through your patient's emotions. Address shame, guilt, fear, and misconceptions. There are some patients who don't realize that the age of onset or disease severity can vary even between members of the same family who have the same variant. Also, empower your patient. How can we increase your patient's perceived control? We can't change the fact that they have this genetic condition, but there are ways that we can help them with how they perceive the uncertainty of their risks, how they react to just having this genetic condition, and how they perceive the value of the medical treatment and interventions that are recommended thereafter. We provide medical and support resources. And of course, genetics is a family affair, so we discuss familial implications, like testing at-risk relatives, and we provide a family letter that they can use to distribute. And also for any family members who wish to be potential donors, we recommend genetic testing in them. And we counsel on the recurrence risk based on the inheritance pattern of the gene, and family planning options are discussed. For example, there is an option to not pass on the genetic condition to the offspring using in vitro fertilization and pre-implantation genetic testing.